the humans are in the
No.
so all the genes are found in the humans are always in the form of pairing. So let's check the structure of chromosome. This is the chromosome, and these two genes are found on the chromosome. So the location at the chromosome where the genes are found, we call this place as a locus. So locus is the presence of gene on the chromosome. And we said all the genes are always in the pairing form, so its replication always found at the same in front of this R. Okay. So now we will discuss the gene pool. What is gene pool? So gene pool, all the genes or alleles that are found in the human or in any animals, such as one of you have all the genomes, such as the collection of all genes in the body or all cells is called gene pool. So we can say like that: we have a jar and we will put some stones in it. When it is filled with the stones, so all the stones may be like in the same shape, so same type. Similarly, all the genes that are found in the human body are called genome or gene pool. So now we will discuss the laws or predictions that we will cover by the Mendelian's genetics. Who was Mendel and what did in the genetics? So Mendel was a priest and he was known as the father of genetics. He did two experiment on the plant. He took pea plant for genetics study. Why he study only pea plant? There are some reasons, because the lifetime or the cross fertilization period of the pea is very small. The other reason, the pea plant can be cross fertilized and self fertilized as well. These are the some reasons why he chose the pea plant. So we will discuss first law of Mendel. The first law is about law of segregation. What is the law of segregation? Is actually Mendel said that all the genes that are found in the pairing form, we call this allele, and all the alleles at meiosis stage segregate or separate as a single allele. So we know that one genes come from the father and the one gene come from the mother. So a cell have two alleles like that, R or small r. So the law of segregation state that when at the meiosis stage, the one genes separate from to the other, and the one cell carry R, the other carry small r. And again, these will, when they fertilize, they are combined with each other and form the new cell that will be the R and small r. So this law is called law of segregation. Other law is about law of independent assortment. What is the law of independent assortment? This law states that when we will study the two traits at the same time, when we study two traits at the same time in a organism, this is called law of independent assortment, and this is also called as dihybrid cross. While in the law of segregation, we study only one character. In law of independent assortment, we study two characters of a organism at the same time. So what is a dihybrid cross? Di means two, and hybrid means an organism that have two combined genes, that we say that R and Y. The R represent color and the Y height. So these two are different character of a person or any organism. So the cross in which we study two types of or two character of any organism, this is called dihybrid cross. Let's go with the prediction. When Mendel took the pea plant, he studied two character. So here is the one, maybe the color, and the other is shape. So the shape of any plant may be round or maybe wrinkled. This is a wrinkled shaped, and this is round shape. He represented the round shape with capital R and the wrinkled with small r. Similarly, he represented the color. The wrinkled color was green and the round color was yellow. So we said there are two traits. These are two traits, and we will study these two traits at the same time. In any organism, the genes may be like R, and Y, Y. These capital, this green color will be represented by the capital Y and yellow with the small y. So we said genes always exist in the pairing form. So these always, this may be R, capital R or small r, and capital Y and small y. This could be R, and Y, Y. This can be also as R, and Y, and y, and this can also be as r and y and y. These are the possibilities of gene or gene prediction. So we cross two of them. Let's start with the capital of all of these. These are, this is one gamete and this is second gamete. According to the law of segregation, every genes come single. So there, from it only we will get R and these are. From this, we will get R and Y. When we cross both of these, we will get R and Y. So this will show us the animal will have round shape and its color will be yellow. Okay. So let's start with the another example such that like R and Y, and here will be the R and Y. So here we have different types of alleles in a single organism. So these will be the, this will not be like similarly as R and R. According to the Mendel, every genes come independently from its parents. So here we get R with capital Y and R with this small y, and this small r will come with the capital Y and this r come with uh, small y. So we will get R and Y, R, Y, R, Y and R, small r. These some gametes that will form through these gametes through meiosis, and similarly this type of gametes will form from this cell such like R capital Y, R small y, small r capital Y, and small r small y. So how can we study these all in a single frame? We have to make a Punnett square diagram here. So all these one gametes we will place at the horizontal, vertical, and these from are placed with horizontal surface. Let's make the boxes. So place every in front of R capital Y, capital R, small y, small r, capital Y, small r, and small y. These are the gametes in front of each boxes. Similarly, these come with in front of this R, Y, R, small y, R, capital Y, and R, small y. So how we cross all of these? First of all, we'll gametes with this, then with this, and similarly with the all of these. So first of all, we will cross fertilize both of these, and the result will be R, Y, and Y. So this cell will be the round and yellow. Now this cell will cross fertilize with this, and the result will be capital R, capital R, capital Y, and small y. So check out there. This cell will be the round and will be the green. But here is a possibility when it fertilized with another cell, this can form a green color. Here, check out R capital Y when cross fertilized with this, but we will get R small r, capital Y, and capital Y. Here is also a possibility that another cell will get
I'm already in the meeting.
okay. So the last cell will fertilize with this, and we will get capital R, small r, and capital Y, and small y. So check here is a more possibilities of a next generation to get this small genes. Similarly, we will cross all of these in the same pattern, and somehow some of these may be wrinkled green, and some of these may be wrinkled yellow. So here is a, I will show you through a picture. Check out there, we have these type of gametes here, and similarly these type of gametes here. Some of them get deviated from their parent characters. So it showed that when the mental cross fertilized, it showed that every genes when fertilized with another genes, it do not affect the genes of other. These are all the possibilities of gene prediction. So R, similarly we will cross this R, R, Y, Y, R. So check out there, we will get round shape, but our color will be the, we will say the color of the seed will be green here, and here R, small r, capital Y, by the small y, capital R, small r, small y, small y. Here also the shape of the seed will be round, but its color will be the green. Similarly, all of these will get the same result, but we will get 9.331 ratio. In all of these, the 9 will be the round and green, while the 3 may be round and wrinkled or green. 3 may be the round and yellow, and 1 will be the wrinkled and yellow possibilities. These are all the possibilities that Mendel checked in the pea plant in the form of their shape and in the form of their color. So, is there any questions about this?
Thank you, Professor Elias. That's very clear. Guys, if you have any questions about this, feel free to to ask. It's okay. There's no bad questions. So feel free to ask if you have any questions. Thank you. Okay, Professor, thank you. If you want, we can move forward to the next topic that you want to cover today.
Okay.
So
the
far,
next
so good.
topic, okay, the next topic is about the chemistry, and we will discuss here some radicals. What are actually radicals? Some elements that may have odd number of electrons. Odd number of electrons that are called radicals, like that chlorine. Its atomic number is 17, and according to the electronic configurations, it is a nucleus, and first of shell will get 2 electron, while the second shell will get 8 electron, and the third shell have 7 electron. So all the elements that have odd number of electrons, we will call them radicals. What is difference between the radicals and negative charge containing elements? We call them as anion. So radicals is a natural form of the element that it may exist in which, while the anion is an element that get electron, or it may form after accepting an electron. So this is the basic difference between radical and anion. Radical is a natural form of an element, while the anion is an element that will get the electron from the another element. So what are the next topic? The next topic is about hydrocarbons. What are hydrocarbons? Hydro means hydrogen, and carbon mean carbon. All the compounds that will contain hydrogen and carbons, we call them hydrocarbons. And in hydrocarbons, the tetravalency of carbon will be 4. The carbon will form 4 bonds in the hydrocarbon compounds. Now we will discuss classification of hydrocarbons. They may be open chain, and they may be closed chain. Open chain hydrocarbons are those also called as aliphatic hydrocarbons. And these two may be as single chain and branched chain. If a hydrocarbon in the form of such like as this will be the single chain, yeah, or open chain. chain hydrocarbon, or we will call this aliphatic hydrocarbon. If there may be a chain in the hydrocarbon or side chain such as this will be the branched hydrocarbon. While these are open from both sides, their all corner are open, so this we call them open chain hydrocarbons. What are closed chain hydrocarbons? The hydrocarbons that are in the form of ring. The hydrocarbons that are in the form of rings or may be closed, also called as closed hydrocarbons, and they may be homocyclic or hetero. Homocyclic, homo means same, and cyclic means cycle, that is may be round. The homocyclic compounds are the compounds in which all the carbon atoms are same, such like as we have carbon, 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 and carbon. These are homocyclic hydrocarbons. And what are heterocycle? Hetero means different. And the cyclic mean in the cycle, closed ring. They maybe are in the form of closed ring, but the atoms maybe are different, such as carbon, carbon, oxygen, carbon, and some like as one of the element in the ring may be different. So homocyclic are those that have same atoms in a cycle compound, and heterocycles are those that contain different types of atom in the cyclic compound. The heterocyclic compounds are maybe as aromatic. That's called as benzene. Benzene is an homocyclic compound. While if any atom in the benzene changed, this will form the heterocyclic compound, such as this is the closed ring of carbon atoms, but here is a oxygen atom. So this is aromatic, but it is heterocyclic with two types of atoms, such like we may have an example of boron, such as ammonium ion is attached at the bottom side of. So in this ring, there are carbon and oxygen, and here is carbon and ammonia ion. So these are some examples of hydrocarbons and their classifications. Now the topic is isomerism. Iso mean same, and merism is mean merging of elements. So any compound that have same type of atom but are different with their structures are called isomerisms. All those compounds that have same formula, that have same structural formula, but are different in their structure, such as we may represent this this compound as in the form of branch, such as CH3, 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 and this branch can this atom can be located at this. So this will be the class. Okay, so all of these have same molecular formula, but they are different in their structure. This is a single chain and this is a branch chain. There are some conditions with isomerism, and how we can say that an element have isomeric formula. For this, the hydrocarbons which have more than 3 number of carbon atom, or we may say that the isomerisms start with the carbon number 4. All those compounds that have more than 3 number of carbon atoms will form isomerism. So we take an example of C5H12, and we will form different isomers of this compound. First of all, we will represent this in the single form. 1, 2, 3, 4, 5. This is one of the form of this compound. We may represent this in another form, that will be CHCHCH.
Está en la mitad de la clase, pero no entiendo nada. Jeremy.
There's 3. And we may take this compound in this direction. So complete the valency of all other atoms, CH2, and here we will get just 1 hydrogen atom. So count them, 3 hydrogen, 2, 5, 3, 3 may form 6, and 3, 2, 5, and 1, 6. These compound have 12 hydrogen. These also have 12 hydrogen, and these have 5 carbon, also 5 carbon, and this is also have 5 carbon and 12 hydrogen atom. So this is a single showing the open chain, while this is showing the branch chain. This can also be represented in the form of we may take this hydro compound to this side. So there is an other compound that have same compound formula, but they have different structure. So we can say that all the compound that have same molecular formula, but we can represent them in different structure, are also called as isomerism. There are some types of isomerisms. The first one is structural isomerism. What is structural isomerism? Basically, the representations of all the compounds in different forms that have same molecular formula is called as structural isomerism, and this may represented in the different form. The first form is chain isomerism. The other one is positional isomerism. The other is functional group isomerism. The other ones are metamer isomerism. And the last one is tautomerism. Now we will discuss all of these in a small details. What is chain isomerism? The chain isomerism represent the compound that have same molecular formula, but we can represent this in the different structure. So take this example as this is an pentane compound containing 5 carbon and 12 hydrogen. This can also be represented as such as this is open chain and this is a branch chain. So chain isomerism means the the compound that can be represented in the form different structures. This will call as chain isomerism. What is position isomerism? Position isomerism means let us have an example of so this is an example of position isomerism. This is a functional group and we can represent this element at all of these positions. This can be attached to here, here, and here also. CH3, CH, chlorine, CH2, CH2, and CH3. This is a compound that have same molecular formula, but we represent the direction of or position of chlorine at different position. So this is a position isomerism. Now next is the functional group isomerism. The functional group isomerism says that the two compounds that have different functional groups but are having a same let's take an example of alcohol. This is C3H8 and here is an example of oxygen. This is a compound and this will represent different functional group. So oxygen, carbon, carbon, so we have total number of 1, 2, 3, 4. Sorry, I count. I take this more. Okay. 1, 2, 3. 3 carbon atom and 8 hydrogen. 3, 2, and 3. These are our carbon atoms. 3, 2, and 3 makes 8 hydrogen atom and also hydrogen atom and 3 carbon 1, 2, 3. While the oxygen atom at the center position representing here the ether groups, while in this form this is representing as a alcohol. So 1, 2, 3, 3 number of carbon atom, and 3, 2, 7, and this hydrogen 8. This is representing here alcohol. So functional group isomerism said that molecules that have different functional groups but have same molecular formula is called as functional group isomerism. Now we will discuss the next is metamerism. What is metamerism? This say that any compound that have same molecular formula but can be we can change the position of functional group at between the hydrogen atom, such as we take example of ester group. Okay, this carbon atom or this functional group can also be represented here. So first of all, we count them 1, 2, 3, and 4. So another structure of this compound is like this. So here we represent this functional group on this carbon. So metamerism means any compound that have same molecular formula but they are different in their structure with the position of functional group on the carbon atoms. And the last is an teutomerism. This is a functional group said that we have this ammonia group. So this is amino acid, and here this hydrogen can also be given to this ammonia group. What will make the loss of hydrogen will create a positive charge here, and here it will get negative charge. So this will be the reversible reaction, and this will form as carbon C double bond negative charge, positive charge, and here hydrogen and NH3 negative with R any functional group. So this is one of our amino acid. When this hydrogen atom we take this to the amine group, this will form as positive charge here and the negative charge on the ammonia. Why these charges are? Because ammonia is getting hydrogen and this carboxylic group losing hydrogen atom. So here is a positive charge. The isomerisms that can be represented in the form of reversible reaction, and they can take hydrogen atom and lose their any atom. We will call them teutomerism. The last example of isomerism is cis and trans isomerism. What is meant by the cis and trans isomerism? These represent the compounds that have similar molecular formula but can be with the different with the representation of their atoms in the space. What is space? We all have studied about the 3D x, y, and z axis. So this isomerism represent the compound in the three-directional forms, such as we take an example of ethene, CH2, ethene, CH, and with chlorine atom. Here is an any compound, and this can be represented as CH. There is a double bond, and CH with the chlorine. So this compound showing us an structure, and we can also show this in the form of carbon, carbon. This chlorine can be represented here, and this hydrogen here. Sorry. And H and H. So this is the cis form, while this is the trans form. This is a single compound or molecule that have structure like this, and we may show it in the form of different space. We can change the position of chlorine from here to here, and from and hydrogen will come down here. So we will get two forms of ethyl chloride, ethene 1 chloride. So if we get the IUPAC name of this compound, this will be the 1,2, and we represent this 1-chloroethene. And similarly for this. So these are the isomerism, and here is a concept about like cis and trans isomerism is only possible for single and double bonded carbon, while it is not applicable to three carbon, three bonded carbon. Why this is so? This is because when we convert the place of chlorine from down to upper, there is a energy change between this bond. This is one sigma bond and one is pi bond. So to break this bond, a huge amount of energy is required. So the three bonded carbon will not form cis and trans isomerism, while the single rotation of the element is easy, and with the double bonded carbon it is also easy, because single pi bond does not require too much energy, and while here is a 2 pi bonds, and this will close the atom more to each other. So 2 pi bonds does not show cis and trans isomerism. That's the overall topic. Is there any question about the cis and trans isomerism or total isomerisms? Is there any question about the topic?
Hello.
Hello.
Yes, so someone have any question about what the professor just explained? No. Okay, so I think we can continue, or we can we have another topic pending, right, Professor?
Yeah, okay, thanks. So about physics, we have topic matter in motion. What actually motion is? Any subject or any body that cover a distance or maybe in a motion have two things. The one is velocity and acceleration. Why motion exists? The motion is due to the, or maybe the result of some force. If someone pushing you, you change your position from one place to other. So this comes in the form of velocity and acceleration. What velocity is and what acceleration is? The change in displacement of body with respect to time. Any body that change its displacement with respect to time is called velocity. And we can represent its graph in the form of y and x axis, and velocity can be of maybe any type. We can say that variable velocity, uniform velocity. So what does mean by a variable velocity? Variable velocity means if any body covering a distance, so this is one position A and the other position is B. If a body is moving with 10 meter per second speed and cover a 10-meter distance in just 1 second of every patches, this will show us uniform velocity. While if it cover 10 meter in 1 second and 15 meter in 2 seconds and 20 meter in another second, this will form variable velocity. The graph of uniform velocity will be like as this, because the speed of the body or the distance that is covering remain constant. The distance or displacement always remain constant, while the time changes at every patch. While with the variable velocity, the body may show this kind of graph, and this can be shown in the form of slope, and the slope will be y over x. The y axis our shows us here displacement, while the x shows us the time. So we say that change in displacement of any body with respect to time is called velocity. So the slope of displacement time graph give us velocity, and this is the change in displacement. So we represent this in the delta d, or delta d means df d initial. Or similarly, change in time occur the time 2 minus t1. So we can find our uniform and variable velocity with this formula. And what is acceleration? So change in velocity with respect to time called as acceleration. Similarly, acceleration can be of two types. It may be uniform or it may be variable. If a body moving with the 10 meter per second speeds at every second, so this will show us a uniform acceleration, and the uniform acceleration can be represented as in the form of straight line. This is y-axis and this is x-axis. So here we can say that if a body is moving with 10 meter per second in every patch with a time t1 in the 1 second, in the 2 second, it is covering 20 and 30 meter in the 3rd second. So acceleration will be the vf minus vi and t2 minus t1. So from these 2 points, we will take 2 points from here. One from the last final velocity, that will be 30 meter per second, and this will be the initial minus 20 meter per second, and the time similarly we take time from both of these last 3 minus 2. So acceleration will be 30 minus 20, 10 meter per second will be the acceleration. So at every patch, the body is moving only with the acceleration of 10 meter per second. So we will call the body is moving with the uniform velocity. While if it covering similarly it is moving with a different velocity, we can say that in the patches A and B, this patch of 10 meter, this is of 20, 25 meter, and this is of 35. So the body is moving with the first distance it cover with the 10 meter per second, this other cover with the 25 meter per second, and the last one cover with the 35 meter per second speed. So here the velocity of the body is not remaining same. So we will say that the body is moving with the variable velocity. Similarly, the graph here represent the slope like y over x represent acceleration. The slope of velocity over time graph show us acceleration. There is another acceleration that is due to the acceleration due to gravity. And we said represent this in the form of g, and these always remain constant at every place. Galileo did many experiments on the towers, and he showed that if you drop down any body from this height in a vacuum, this will come with the same acceleration to the earth, or that will be 9.8 meter per second square. If you drop an elephant from here, it will also take the same time as if we drop a leaf of a tree from this place. Both will take same time and cover and come with the same acceleration, that will be 9.8 meter per second square. So there are some equations that represent the motions of a body in a straight line. First of all, that will be the Vf minus Vi plus At. The second one is of distance, that will be Vi plus 1/2 At square, and the last one is 2As equal to Vf square minus Vi square. These all have an acceleration constant that representing the body that moves in a straight line motion. When we discuss about the straight line motion or uniform gravity or uniform motion, then we will use these equations for numerical process. If somebody have moving with the velocity of 20 meter per second and have an acceleration of 4 meter per second square, so what will be the time? We can solve this by simply, as the data is Vf 20 meter per second, while the initial velocity 0 meter per second, and acceleration equal to 4 meter per second square, while we have to find the t. Simply put this all data into the equation and solve for the t, just as Vf equal to 20 meter per second, initial velocity of the body 0, while acceleration we have 4 meter per second, and we have to find the time here. So this 4 is multiplying with the t, and when we drop down here, this will come in the denominator, and the answer will be 5 meter or 5 second. This means the time that a body totally covering with the velocity of 20 meter per second have time 5 seconds. So also, if we want to find the distance of a body that is moving linearly, these all terms for the same motions: linear motion, uniform gravity, or straight line motion. We always use these type of equations to solve the numerical. So the next topic is about force, and we know that this is given by the Newton. there are three laws by the Newton to define the forces. The first law is saying that every body that remain in constant motion always remain in the motion, and every body that is in the that is at some place and not moving, that continue their motion in a constant way. So the first law say that every body that moving with some uniform motion always remain in continuous motion until an external force disturb it. Until the external force disturb, the body always remain in the constant motion. This is the first law, and the first law of Newton define force. First law define forces, while the second law give us knowledge about the acceleration. The Newton said that if we apply a force on any object or any body, it continue motion with some acceleration. The force produce motion in the mass or in any object, that will be the acceleration. The force is directly proportional to the acceleration, and the force will be directly proportional to the mass. So here we can say that if we are given with the mass and force, we can find the acceleration to find the acceleration of a body. Let's show us here an example. If you apply a force of 25 Newton on a brick that have mass of 5 kg. So what will be the acceleration? That will be 25 Newton equal to mass 5 kg into acceleration. This come in the denominator, and we will get acceleration of 5 meter per second square. So the second law of Newton tell us about the quantity of force. The first law define force, while the second law tell us about the quantity of force that can be applied on the body, or it may tell us about the acceleration. While there is a third law, that is every action has same reaction. If you hit somebody with the force of 10 Newton, the same force will be applied on you by the body. That is the action reaction force. And action reaction force, and for action reaction force, there will be two bodies. For action and reaction, there will be two bodies. Action and reaction cannot be applied on the same body. If you are applying a force on the body, then the body applied force on you. So every action has same and same reaction. We can say that if you are finding an data with the numerical calculation, if someone says that you have force of 200 Newton that you are applying on the body, so what will be the reaction force? This is the action force, so the reaction force will be minus 200 Newton. Look here, you are applying a force in this direction, while this body applied force on this direction. So action and reaction always in the form of anti-parallel. Anti-parallel form, or we can say that what is the angle between action and reaction forces, or that will be 180 degree. Someone applying in this direction, then the body will apply on the same direction toward it. So there will be 180 degree angle between the action and reaction forces. These all about the forces and matter in the motion. If you have topic about 2D projectile motion, you can tell me. Projectile motion in the same chapter. Can someone tell me? Can someone let me know
Paul?
about the
Paul, would you like to take a crack at that question?
are you asking me?
No, I was asking one of our students, Paul, if he wanted to take a crack at the question. Can you repeat the question, Professor?
Can someone let me know about the topics in the chapters? Is projectile motion is in the same chapter?
Oh, yes, it's fine. I think for this topic, I wanted to share with our students and with you is all of the topics, the three topics we covered today in biology, chemistry, and physics, they're very specific topics to the lessons in the course. So the idea is these are the lessons, Professor Elias and Paul, where most of our students send us questions, or when we do the online quizzes, they have questions about it, or sometimes we need to do extra work. So the topics that you covered are the most, I guess, challenging, or the ones that have the most questions from students, but they are very, very in-depth. So it's hard to cover them independently. They're part of the, the course. Usually you take several lessons in order to get to the topic that we're covering today. But the idea of these tutoring sessions with you, Professor Elias, is to support the most challenging part of the curriculum. So this is our chance, Paul, if you have any questions, or Aguilar, if you have any questions, please feel free to ask, and we'll be happy to interact with you here live. So as I mentioned before, there's no bad questions, so feel free to ask us now, or to send us the question via the platform if you want later. It's up to you.
If anyone have questions, you can ask me.
Daritza, is there anything else?
I think we're good for now. If no one has any questions, I think we covered already the 3 topics for today's lesson.
Okay, well, Professor, I thank you very much for your time today and for the extra time. I think we went a little bit over the scheduled time, but I think all of our students here were very interested. Everybody stayed on. So, I mean, most of the students who started are still connected. So I think you kept them interested. Thank you, Professor Elias.
Okay. Okay. Thank you to all.
Oh, can we see you for a second so we can say goodbye?
Okay.
Okay, fantastic. Thank you,
Okay.
Professor.
Thank you.
I appreciate it. Thank you for your time.
Okay.
All right.
Okay. Thank you.
Ciao.
Thank you everyone for joining.